we're going to go ahead and try to get this next lesson knocked out of the way on cell structure and function. It's going to be a bit of a long one, so brace yourselves. You may have to watch it in chunks. So we're going to go through basically all of the various organelles that are found in cells. And first, I wanted to focus on some of the organelles that are found in all cells on the planet. Ribosomes, for instance, are found in every cell. They are not surrounded by a membrane, so they're not membrane-bound organelles. They're honestly very tiny. They run the border between a very large molecule and a very tiny organelle. They're made of RNA and protein. So in this diagram, the orange and the yellow that you see is actually ribosomal RNA or RNA. And then the purple that you see is protein. So they're kind of a hybrid molecule as well. They are found in all cells on the planet. So they are kind of some of the evidence of the common ancestry of all life, all organisms. They can be found in one of two locations. So any given ribosome could be found floating free in the cytoplasm or attached to the rough endoplasmic reticulum, which we'll talk about later. And it kind of depends on which particular protein is being made. So you could have a ribosome that at 1 minute, depending on the protein it's making, is floating around in the cytoplasm. And then if it has the instructions from an mRNA, if it has an mRNA attached with a different type of protein, it could then attach itself to the endoplasmic reticulum and start pumping out that different protein. Then moving on, we have cytoplasm. So this is the contents of the cell, everything in the cell except for the nucleus. It has two components, the cytosol, which is the fluid that kind of surrounds all the organelles, and then the organelles that are stuck in it. Also contains the free-floating ribosomes, and you have some important chemical reactions happen there. For instance, glycolysis, which we'll talk more about when we talk about cell respiration. So in this diagram, here's my plasma membrane. My nucleus is for some reason not showing up, but the nucleus would be right here. And sorry, I'm going back and forth between my computer and my tablet, and for some reason, some things aren't showing up on the tablet. I wanted to point out that the cytoplasm and, trying to get to the next slide, and the cytoskeleton are not explicitly named in the course and exam description, but they're pretty important for us to be able to talk about some of the things that we're going to talk about later, especially since we don't cover the cytoskeleton in freshman biology, so I wanted to make sure that I brought it up. So the cytoskeleton is, like its name suggests, a skeleton for the cell. It's going to do a lot of functions for a cell that a skeleton does for your body, for example. It's made of mostly protein fibers and filaments. Filament means thread. There are 3 different types of proteins. You don't need to know a lot about each of these different protein types. What you need to know is that the cytoskeleton helps to anchor organelles in place, helps the cell keep its shape. It allows cells to move, so any cells that move on their own, it's the cytoskeleton that's helping them move, and it also allows organelles to move within a cell as well. Then microtubules are an important component of the cytoskeleton. They're going to come up again when we talk about mitosis and meiosis, and we're going to talk about the microtubules involved with centrioles and spindle fibers. The plasma membrane or cell membrane we're going to talk about in its own specific lesson, lesson 2.3. So I'm just mentioning it here. It's found in every cell on the planet, and we're going to talk about it later. A similar organelle, the cell wall, we're going to talk about in a different lesson as well, so I'm not even bringing it up here. Then we have what's called the endomembrane system. So this is going to include a lot of the organelles that we tend to talk about that you may not have known are part of a similar system. What is the endomembrane system? Well, it's this series of organelles that are surrounded by membranes. And the important thing is their membranes can be interconnected. So what you see here is basically a folding in of the membrane. And that membrane folding in can pinch off and form these little, what are called vesicles. And those vesicles can take that bit of membrane and anything that's contained in it and can move between these different organelles. So the organelles that make up the endomembrane system are the endoplasmic reticulum, or ER, the Golgi complex, lysosomes, vacuoles, transport vesicles, the nuclear envelope, and the plasma membrane. So you could theoretically have a molecule that's embedded in the plasma membrane eventually find its way to like the ER. It would be carried by one of these little vesicles. That's going to be carrying membrane from piece to piece of the endomembrane system. And then these structure of this is that it's interconnected, and the function has to do with the fact that this is all about modifying and packaging various molecules like polysaccharides. Remember, those are carbohydrate polymers, various lipids and proteins, both between cells themselves. So you could have something that's produced here in the ER. It could be carried out to the Golgi, and then from the Golgi, it could go to the plasma membrane, and from the plasma membrane be excreted or released from the cell. It's also, this endomembrane system transports things within the cell as well. The endoplasmic reticulum is physically interconnected with the nuclear envelope or nuclear membrane. The nucleus we don't talk about separately, but the nucleus does contain the DNA. We'll talk about it in a little more detail when we talk about, say, mitosis, because that, and also the structure of DNA, because that kind of relates to the nucleus. Endoplasmic reticulum is important for intracellular transport. Intra means within, so this is inside the cell. Then there are two types of ER. Rough ER has membrane-bound ribosomes attached to it, but these ribosomes, remember, can come on and attach to the rough ER or pop off if they're creating some protein that is not going to be associated with the ER. And the ER has all these compartments. In these different compartments, you can modify some of these proteins that are being made. It allows for you to have slightly different physical conditions than you might have in the rest of the cell. So you could have different pH. You could concentrate some proteins so that they'd be able to work on whatever you're trying to modify. And then protein synthesis and modification happens here. Then you have the smooth ER. Its role is going to be in synthesizing lipids and steroid hormones, as well as playing a role in detoxification. In general, if you're looking at cells and they have a large amount of smooth ER, they may have a very heavy role in something like synthesizing these lipids or steroids or detoxifying. So liver cells, for instance, are going to have large amounts of smooth ER in them. The Golgi complex, you will see, has several different names. Sometimes you call it Golgi apparatus. Sometimes it's called Golgi body. In our course and exam description, it's called Golgi complex. Sometimes I'll just say Golgi so you don't get confused, or I'll call it the Golgi ABCs. The important thing to note is what its structure and its function is. So it's a series of flattened membrane stacks. I think it looks like very messy pancakes stacked up. And the function of it is you're going to have proteins that are made from the ER. They're going to come into, if you ever see it, something called the cis face, which is facing the inside of the cell. You have these incoming transport vesicles. They will melt into one of these sacs, and then these transport vesicles can also carry materials from one of these sacs to another. And in each of these sacs, they will be modified in one way or another to help fold those proteins, maybe add some chemical modifications to change their function a little. And then finally, they're packaged for export from the cell. And that's going to happen from the transface. Transface means opposite. So it's going to be facing the cell membrane. So the plasma membrane would be out here. And then you have, again, these transport vesicles that are going to then take whatever materials have been modified in the Golgi, and they're going to take them to the plasma membrane so they can be excreted or released. Okay, lysosomes are little balls of membrane that contain digestive enzymes, otherwise known as hydrolytic enzymes. And keep in mind that hydrolytic refers to enzymes that perform hydrolysis. So they're going to be breaking materials down. These are found in mostly animal cells, some fungi and protocells as well. They're not found in prokaryotes or in plant cells for the most part. They can also play a role in apoptosis, which is programmed cell death. So the digestive enzymes in them can be released into the cell to help start breaking it down if the cell needs to basically be sacrificed for the rest of the organism to survive. Then you have vacuoles. So vacuoles are also balls of membrane, but they can do different functions. So in plant cells, they're especially important. You have a large central vacuole, and that central vacuole is going to be important for something called turgor pressure, which helps maintain the shape of a plant cell. We'll talk about that a little bit later in this unit. You also store water and nutrients and ions in the central vacuole. It is a very large structure in the plant cell. cell. It's often one of the largest structures in a plant cell. And in animal cells, there are smaller vacuoles. You may have more than one vacuole, and they're used mostly for storage. Then we have another category of organelles that are going to become a little later on when we talk about how these complex cell structures evolved. I'm calling them semi-autonomous double-membraned organelles, which sounds very fancy, but what does that mean? Well, these organelles contain their own DNA and they have their own ribosomes, so they do some protein synthesis on their own. They are surrounded by two membranes. That's the double membrane aspect of it. And the semi-autonomous means they're slightly kind of independent of the rest of the cell. So they, for instance, reproduce and replicate independently of the rest of the cell. You have cell division happening, and then somewhere within that cell cycle, you may have mitochondria or chloroplasts just reproducing or replicating on their own. So I already mentioned those two types of organelles. You have mitochondria, and this diagram is a little more detailed than a lot of the ones that you'll normally see because I wanted to make sure you could see that it contains its own DNA. It has ribosomes. The double membranes that you have an inner membrane and an outer membrane. We'll talk more about the significance of the double membranes in a little bit, but another thing that the double membrane allows for is compartmentalization. What that means is that you've got these different compartments where you can have separate materials, you can have different pHs, you can have different concentrations of various chemicals, and that's really important. We're going to talk about that more when we talk about cell respiration. The outer membrane is smooth. The inner membrane is highly folded, and we're going to talk more about that when we talk about membranes and their properties, because when you have a highly folded membrane, that's usually trying to create more surface area because you have something in that membrane that is doing a job and you want to have more of it. So this allows for more efficient ATP synthesis because mitochondria are involved in cellular respiration. Keep in mind it's aerobic cellular respiration, which means with oxygen, and cell respiration means it's going to make ATP from glucose or other carbohydrates. Then, speaking of glucose, it is produced in chloroplasts. Chloroplasts are found in plants, cells, and photosynthetic algae. So it is the location where photosynthesis happens in a cell. Notice it also has an outer membrane and an inner membrane. We're going to talk about the structure of a chloroplast in more detail when we get to photosynthesis. I just wanted to point out that it also has its own ribosomes and it also has its own DNA that's separate from the DNA in the nucleus of the cell. And having these compartments or these separate membranes are going to allow chloroplasts to, there's basically the chemicals, the molecules and proteins that are involved in photosynthesis are found on this inner membrane, and having a highly folded inner membrane means you've got more space to carry these enzymes and proteins. All right, so that's all we're going to talk about for now, and we've got a lot more detail about cells than you probably ever wanted to know coming up in the next lessons.